Hello students, welcome back to biology revision class for grade 12. Today's topic is cell biology from grade 11 unit 4. Stay tuned. Students, pay attentive to this revision class, and the revision is begin with the history of cell biology, cell theory, and investigate the size, structure, and shape of the cell, basic functions of the cells. All life on Earth originates from the cells. So these are the important points to be revised. So now let's begin with the first part of this cell theory. Before scientists tell us about the cell, they have developed different kinds of theories. So here, the development of modern cell theory, living things are made of cells. It explains billions of cells in the case of human beings. Just one cell in the case of organisms like amoeba. So you can see in this slide, there are human cells, different blood cells, nerve cells, muscle cells, and in a single cell also here in this slide, the amoeba cell. And there is only one cell in amoeba, paramecium and vorticella. Only a few hundred years ago, cells had not been discovered. The discovery had to wait for the development of the reliable microscope to magnify sufficiently to solve the cellular structure of living organisms. Many biologists and other scientists contributed to the discovery of the cell, and the statement of the very first cell theory came into being. So here you can see that in this slide, the amoeba, the paramecium, the vorticella are some of the single-celled or one-cell organisms. So here you're gonna look again the timeline for the development of the cell theory. Here, how cell theories have been developed. There are many process goes on in this development of cell theory, beginning from 1665 with the invention of the crude microscope with the Robert Hooke, with one of the earliest compound microscope. As you can see here in this slide, makes drawings of cork and see tiny structures that he calls cells. The lenses are not very good, and magnification for more than 30 times are very blurred and do not show much detail, so only dead cells. So this is the first beginning of the development of cell theory, with the development of small crude microscope with Robert Hooke. So he only observed dead cells in his microscope. Okay, the living cells are not observed because he took cork cell from the tree bark, dead tree plants. Again, in next coming in 1674, Anton van Leeuwenhoek see living, moving unicellular organism that he calls protoctistan in a drop of water. He is using a simple microscope with only one lens. It is really little more than a magnifying glass with a mount for the specimen. His microscope can achieve magnification of 300 times, which is more than Robert Hooke microscope, that is 30 times. He calls the moving organism as animalcules. He also see bacteria from his thesis, which he also called tiny manicles. So Robert Hooke also is coming in the second place in the contribution of the development of cell theory with his crude microscope. And gain insert in 1824, French biologist Rene de Trochette conclude that all organisms are composed of cells. This follows many years' work in which he also discovers the stomata and the epidermis of the leaves, the process of osmosis. Chlorophyll is needed to photosynthesis to occur. Respiration occurs in both animals and plants. So apart from the contribution to the cell theory, Rene has also discovered many things that we have mentioned earlier. De Trochette is the man who first stated the cell theory by recognizing that all organisms are made up of cells and that all growth occurs because of the increase in the volume of the cell or the addition of more little cells. And again, following Rene de Trochette in 1939, Matthias Schleiden and Theodor Schumann, coming from Germany, put forward the first clearly stated cell theory. It states that the cell is the unit structure, physiology, and organization in living things. The cell retains a dual existence as a distinct entity and a building block in the formation of organisms. The cell formed by free cell formation, which is later disproved this, that is not the cells are free cell formation, that is the idea of spontaneous generation. So these are the main ideas regarding Matthias Schleiden and Theodor Schwann in the course of development of the cell. So here these are the two persons from Germany. Next, in 1858, Rodolph Virchow, a German doctor who develops many surgical techniques and promotes several fields of modern medicines, declares that omni cellula cellulae, which means that a cell can only arise from another cell like it. Why this he completes? With this, he completes the first accepted version of the cell theory. So this is from Germany, and all the organisms are made up of one or more cells, according to Rodolph Virchow. All cells came from pre-existing cells. The cell is the unit of structure, physiology, and organization in living things. The cell retains a dual existence as a distinct entity and a building block in the construction of organisms. Today, to conclude on the development of the cell theory, today this has been modified and extended in the light of our increased knowledge of genetics and cell biology, and now reads all non-living things are made up of cells. The cell is the unit, structural and functional unit of all living things. All cells come from pre-existing cells by division. There is no spontaneous generation of cells. Cells are not come to being with the spontaneous generations. That means non-living things. Organisms are coming from non-living things. This is already disproved. So cells contain hereditary information which is passed from cell to cell during cell division. All cells have basically the same chemical composition. All energy flow, that is the metabolism and biochemistry of life, occur within cells. So these are the part of the line of cell development, and there are also some key events in the study of cell biology. As you can see here, besides these major steps in the development of cell theory, there have been other development in the study of cell biology. Some of these are listed in this table, as you can see in this slide. So here in 1995, Johnson builds the first compound microscope. In 1626, Redi postulates that living things do not arise from spontaneous generation. This is, we have mentioned earlier that how the theory of spontaneous generation disproved by the Redi and Pasteur. So in 1665, Hooke describes cells in cork. In 1674, Leeuwenhoek discovers protozoa. In 1833, Brown describes the cell nucleus in the cells of an orchid. In 1939, Schleiden and Schwann propose a cell theory. In 1857, Kolker describes mitochondria. In 1858, Virchow states omni cellula e celluli. In 1869, Measure isolate DNA. In 1879, Fleming describes chromosome behavior during mitosis. In 1898, Golgi describes the Golgi apparatus in cells. In 1939, the first transmission electron microscope invented. In 1953, Watson and Crick proposes the double helix structure of DNA. In 1965, the first scanning electron microscope invented. And in 2000, human genome DNA sequence drafted. So these are the key events in the course of development of the cell theory. Now we proceed our discussion to the cell size. How big are cells? A chicken's egg are just one huge cell packed with food. It is a pretty big cell, up to 5 centimeter or 0.05 meter in length. The smallest bacteria cell are only just over 100 nanometer in length. This is approximately 100,000th of the size of the chicken's egg. That is quite a range of the size. So the cell ranges from that of the ostrich or the hen eggs to that of the bacteria, the minute structure, the smallest cell. And units used to measure the cell. There are big cells and small cells. In between, there are a range of different cell sizes are there. It depends on, to measure the cell size, it depends on which cell, which unit are available, and which one would be convenient to use. So it all depends on these ideas. That is, which cells, which units, and which one would be convenient to use. Cells can be measured in meters. The size of the cell in meter would be very, very decimal in measurement. So this is not conducive to use like meters to measure cells, rather to use very small units. So all the zeros are very confusing after the decimal, and we don't easily work with such small numbers. Therefore, we use other smaller units to measure the size of the cell and the molecules. Here you have on the slide different sizes of the cell, as you can see. The smaller unit give us numbers that are more convenient to work with. There are 3 small units commonly used: the millimeters, micrometers, and the nanometers. So these are convenient units to measure the size of the cell. And there are figures that are used to measure these different sizes, and you can see on the slide. To convert a larger unit to the next smallest unit, always multiplying by 1000, and for example you want to convert 1.5 millimeter to micrometer, so 1.5 millimeter times 1000, this becomes 1500 micrometer. To convert a smaller unit to the next larger unit, divide by 1000. These are small rules, we can say, that apply in measurement, in conversion of one unit to the other, from smaller to bigger unit, or from bigger unit to smaller unit, vice versa. So here, for example, to convert the smaller unit to the next larger unit, divide by 1000. For example, to convert 90 nanometer, this is a smaller unit, to that of larger micrometer. 90 divided by 1000, this 0.09 micrometer. So this kind of conversions are there in measuring the cell size. So our red blood cells, that was 0.00, there are 5 zeros plus 7 meter in diameter. If we want to measure it in meter, the red blood cells, that is 0.07 in millimeter, or 7 micrometer in diameter. So it becomes to the lowest smallest unit measurement. So it is very easy to apply these small measurements, and the numbers are easily read. The size of the cells can be imagined, how far they are large or small. So smaller units are very preferable than the larger units. This is a much more comprehensible number. Most cells fall within a much narrowed range of size than the chicken's egg and the smallest bacterium. The lengths of most animal and plant cells fall with a range of 10 micrometer to 100 micrometer. Most bacteria are about 1/10 of these lengths. The relative size of the animal cell, the bacterium, and the virus is a slightly different way. The diagram makes it clear just how much bigger an animal cell than a bacterium. The animal cell may be just 10 times as long, but it is also 10 times as wide and 10 times as deep. This makes it 1000 times bigger than bacterium cell when we compare bacterium and animal in this slide. So consequences of the different size of the cell. So what are the consequences, the outcomes? Having smaller size, having bigger size, or medium size. When a cell gets bigger, all its dimension change. When a cell doubles all its dimension, it is twice as big. But in fact, there is now 8 times more cells as a result. This is most easily explained if we present that our cell is a cube. But the same principle holds true for other shapes also. So here we can use the cell in the cube size. The cube cells of different sizes. The linear dimension double from the first cell to the second, double again is from the second cell to the third. But the surface area and the volume of the cell does not double. So this is the cube that you can see in this slide. The smaller, the medium, and the largest volume of the cell. So as the cell increases, the surface area to volume ratio decreases. So this is related with their size of the cell. There are 6 sides to a cube. Calculate the area of each side, multiplying the lengths by breadths. In the case of the first cell, that is 1 unit arbitrary unit. So the area of one side is 1 arbitrary unit square. The volume of a cube is length times breadth times 8. In this case, 1 arbitrary unit times 1 arbitrary unit. So the volume of the first cell is 1 arbitrary unit cube. So the ratio of the surface area to volume ratio is 6:1 in the first cell cube. And so that the second cell, what about the second cell? The linear dimension have doubled, but the surface area is 24 arbitrary units square and the volume is 8 arbitrary cube. The ratio of the surface area to volume ratio is 24 divided by 8. This gives you 3:1. So it has half of that of the small cell here. As the cell size increases, surface area to volume ratio decreases. This is in the second cell. The ratio of the surface area to the volume of the third cell is in fact 1:5:1. So it's very much small again. Surface area to volume ratio. The first cell is the bigger one in its surface area to volume ratio as compared to the other two cells in the two cube cells. Smaller again, and half value of the ratio of the cell with the linear dimension that are half of this one. So the size has an impact with the cell's materials transport and cell's metabolic processes. So surface area to volume ratio impacts rely upon the function of the surface area of the cell and the volume of the cell. It is best understood by considering one function, that of respiration. A cell respires to release energy to drive all other cellular processes that take place. If it can't release enough energy, these other processes slow down and the cell may die. In order to respire, the cell needs oxygen, which enters through the surface of the cell. So surface area to volume matters for material exchange energy releasing mechanisms. The volume determines how much activity there is in a cell. A large cell will have more process happening, or at least the same process happening faster than a smaller cell. The amount of energy that must be released in respiration is therefore decided largely by the volume of the cell. So here you have different kinds of cells, just to show you from different organisms. The amount of oxygen that can be delivered into the cell is decided largely by how much surface there is, since it is through the surface of the cell that the oxygen enters. A large surface area to volume ratio means that it is likely that the surface will be able to supply the oxygen demand of the cell. Here you have different cells, but as the cell increases in size, the volume decreases faster than the surface area, obviously, and the surface area to volume ratio decreases. Here you can see we can measure, see the different sizes of cells with a microscope, except the ostrich egg and the chicken's egg. We can see with the naked eyes. So some cells are seen with the naked eyes. The majority of the cells seen with school compound or light microscope. We have electron microscope, but most conveniently the school microscopes are used for investigating the different cells. So we have come to the end of the first session. And we have some revision questions. Students, listen attentively to these revision questions and answer and look for the correct answer. I mean, you listen to the question and answer the questions that follow. So here the first question is, name of the scientist who proposed the extension of the cell theory is A. Robert Bentley, B. David Bellamy, C. Rudolf Virchow, D. Thomas Bell. Student, if you have answered C, you are correct. Rudolf Virchow is the first scientist who proposed the extension of the cell theory. Second question, the nucleus of the cell was discovered by A. Robert Hooke, B. Robert Brown, C. Aristotle, D. Matthias Schleiden. If your answer is B. Robert Brown, you are correct. Third question, name of the scientist who made the first microscope is A. James Watson, B. Lewin Hooke, C. Eukaryotes Johnson, D. David Baltimore. If your answer is C, you are correct. That is Eukaryotes Johnson. Fourth question, who saw the first cells? A. Hooke, B. Van Leeuwenhoek, C. Schwann, D. Johnson. If your answer is A, you are correct. A. Hooke, Robert Hooke, who saw the first cell with his crude microscope. Question number 5. Which of the following is not one of the main components of the cell theory? A. Cell must contain DNA. B. All living things are made up of cells. C. Cells can only come from other cells. D. Cells are the basic unit of life. So your answer is A, you are correct. Cells must contain DNA. Some cells, they might not have DNA. Like bacteria that contains nucleoid region. They don't have exactly nucleus. So not all cells must contain DNA. So the answer is A. The last question. Which technology was essential for the development of cell theory? A. Telescope. B. Microbes. C. Antiseptics. D. Microscopes. If your answer is D, microscopes, you are correct. This is the first session. Now let's go to the second session of this revision, that is types of cells. How many types of cells are there? Exactly, there are, based on their structural complexity, there are two types of cells, prokaryotic cells and eukaryotic cells. We're going to look at each of these step by step. First, let's consider the prokaryotic cell. First type of cells to be formed when life first evolved, the prokaryotic cells. The prokaryotic cells are much smaller and simpler than eukaryotic cells. Some division of lever within the cell. There are specialized regions for certain functions. So you can see the prokaryotic cells, as an example, is a generalized prokaryotic cell in this slide. It has some various complex structures. You can see the flagellum, the internal section, the outside exterior section of this bacteria cell. So eukaryotic cell is the second division. There is much more division of lever in a eukaryotic cell. There are different types of eukaryotic cell, but all of them have a number of features in common. Most of the structures shown are found in all eukaryotic cells. Plant cells have a number of other structures in addition to these common structures. So here you have a generalized animal cell as well as a generalized plant cell in this slide. There are many more different individual structures called organelles in the cell. Also, there are many more membranes in the cell. Some of these form the complex membrane system that is found throughout the cell, the endoplasmic reticulum. Several of the organelles are surrounded by the membrane. So here we have different structures of the cell, the organelles. These are called membrane-bound organelles, nucleus, mitochondria, chloroplasts, if present, lysosomes, Golgi apparatus. The membrane-bound organelles, cellular reactions not affected by other cellular reactions. So organisms in a given cell, the eukaryotic cell, you have many organelles inside the cytoplasm, and these each organelles are bounded by membrane. So there is no interference with one reaction of the organelle with that of the other one. So the membrane encloses not to interfere with their tasks or cellular functions. Origin of eukaryotic cells. There are some theories and explanation behind the origin of the eukaryotic cell. As we have mentioned earlier, the prokaryotic cells are the first cell appeared on this planet. One theory is that the modern eukaryotic cell was formed when several of the more primitive prokaryotic cells got together. Over millions of years, ancestral prokaryotic cells became more membranous. The plasma membrane around the cell became more and more folded until there was an extensive membrane system in the cell. This would eventually evolve into the endoplasmic reticulum, for short EPR, of the eukaryotic cell. The next stage in the theory suggests that this is membranous cell engulfed the other smaller cell that were better at respiring organic molecules to release energy. These engulfed prokaryotes would evolve into the mitochondria of eukaryotic cells. The cells that contained them were heterotrophic, and the forerunner of the animal, fungal, and protoctistan cell. One further stage suggests that some of these cells were their primitive mitochondria also engulfed other smaller prokaryotic cells. These were prokaryotic cells that would photosynthesize and would in time evolve into chloroplasts. These cells that contained these would be autotrophic and the forerunners of the plant cells. This theory of the origin of eukaryotic cell is called the endosymbiont theory and was first proposed by the biologist Rene de Trochette. Difference between prokaryotic and eukaryotic cells. These two cells do have their own differences. The similarities and similarities are there. So prokaryotics lack an organized nucleus and other membrane-bound organelles. This is the major difference between eukaryotic and prokaryotic cells. They lack nucleus and membrane-bound organelles, the prokaryotic cell I mean. Prokaryotic DNA is found in the central part of the cell called the nucleoid. The cell wall of the prokaryote acts as an extra layer of protection, helps maintain cell shape and prevent dehydration. Prokaryotic cell size ranges from 0.1 to 5 micrometer in diameter. So the major differences between the prokaryotic cell and the eukaryotic cell displayed in this slide on this short table. So students, when you got this, you can go through this table and you can easily understand the difference between prokaryotic and eukaryotic cell in terms of cell, nucleus, DNA, mitochondria, chloroplast, ribosomes, and the like, cell walls including. The small size of prokaryotic allow quick entry and diffusion of ions and molecules to the other parts of the cell, while also allowing fast removal of waste product out of the cell. So being a small cell has very active cellular process. Now when we came to the second cell type, what's the difference? We try to make a distinction between the eukaryotic and prokaryotic cell. So now let's look at the second one. The eukaryotic cell are larger than the prokaryotic cell and have a true nucleus, membrane-bound organelles, and rod-shaped chromosomes. The nucleus houses the cell's DNA and directs the synthesis of protein and ribosomes. Mitochondria are responsible for ATP production. The endoplasmic reticulum modifies proteins and synthesizes lipids. The Golgi apparatus is where sorting of lipid and protein takes place. So these are the main tasks of the eukaryotic cells. Peroxisms carry out oxidation reaction that break down fatty acid and amino acids and detoxify poisons. Vesicles and vacuoles function in storage and transport. Animal cells have a centromere and lysosomes, while plant cells do not. Plant cells have a cell wall, a large central vacuole, chloroplasts, and other specialized plastids, whereas animal cells do not. Students, this is the second part of the lesson, and now we go to the revision question. Here in the revision question, in a similar way, you answer the questions. Question number 1. The unit of measurement used for expressing dimension or size of the cell is A. Centimeter, B. Millimeter, C. Micrometer, D. Meter. So which one is preferable? If your answer is C, you are correct, because centimeter, millimeter, or meter are larger than the micrometer. So the very preferable unit here is the micrometer. So choice C is correct. Second question. Under a microscope, Lulit observes a cell that has a cell wall, but not distinct nucleus. The cell that she observes is A. a plant cell, B. an animal cell, C. a nerve cell, D. a bacteria cell. Your answer is D. You are correct. A bacteria cell. Question number 3. A difference between eukaryotic and prokaryotic cell in having A. ribosomes, B. nuclear membrane, C. cell wall, D. none of the above. In having, the difference between eukaryotic and prokaryotic cell is in having what? The answer is cell wall. Your answer is C. You're correct. Question number 4. What is the name of the region where double-stranded single circular DNA is found in the prokaryotic cell? A. protonucleus, B. nucleoid, D. nucleoplasm. If your answer is B. nucleoid, you are correct. Question number 5. Identify the correct statement. A. Tissue is a group of dissimilar cells. B. An organ consists of similar cells. C. Vacuoles are not found in plant cells. D. Prokaryotes do not have nucleus. The answer for this question is prokaryotes do not have nucleus. D. Students, this is all what we have for today. Till next time, goodbye.